Hello, good evening. Welcoming you to the week 12 live session of experimental biochemistry. I am Surasree. I am a PMRF research fellow in the Department of Biotechnology, IIT Kharagpur. So this is our last live session for this semester, and in this course, but in this particular experimental biochemistry course, we have discussed several topics of biochemistry, several aspects of biochemistry, starting from protein structure, amino acid structures, to the concept of pH. Then we have discussed several protein purification techniques, some preliminary aspects of recombinant DNA technology. And in this last session today, we are going to discuss immunoassay techniques: southern blot, northern blot, and western blot. So let's begin. So the first immunoassay technique that we are going to discuss today is southern blot. And so what is southern blot? These blotting techniques or these immunoassay techniques are important because they are used to detect the biomolecules, like for example DNA, RNA, or protein. So the southern blot technique was developed by Edwin Southern, and on his name it was named as southern blot. And this immunoassay technique is a technique used for detection of specific DNA sequence. So there are several aspects of this experiment. The first one is that we are trying to detect a specific DNA sequence from a DNA sample. So suppose we want to detect if one particular gene is present in our DNA or not, in our organism or not. So what we do is we isolate the organism's genome, and then we allow it to be digested by restriction endonucleases, right? So the restriction endonucleases, they cut the genome in several pieces, and then we run this DNA sample on an agarose gel. So the first step is restriction digestion of the DNA. So what restriction digestion does is that by the help of restriction endonucleases, we can get small pieces of the DNA. We can get small pieces of the DNA or small fragments of the DNA, and then we run these fragments on agarose gel. So gel electrophoresis we had discussed previously. We know that agarose gel is a gel used for running DNA samples mostly, and long DNA samples. So this is one agarose gel where we run the DNA sample, and these DNA fragments will be separated on the gel according to their size, right, according to their molecular weight. Now the next thing will be, so this part is separation. Now the next thing will be to transfer this particular this particular DNA fragments to a membrane. So the next step is transfer onto membrane. Now why is it important, or why is it necessary? So the DNA fragments that we are running on the gel, these DNA fragments could have been directly detected by the by the probe DNA, which we are going to discuss later on. So probe DNA is something which is a specific DNA which is complementary to the DNA that we want to observe, right? And this probe DNA will hybridize with the particular specific DNA of our interest, and then finally this probe DNA will be labeled so that we can visualize this probe DNA, and then we can conclude about the presence or the existence of our particular DNA of interest in the sample, right? But the problem here is that this particular experiment of this detection of the DNA fragment is not possible on the gel. So what we do usually, we transfer the fragments of DNA, or in the later experiments we will see in case of northern blot RNA, or in case of western blot protein. So these DNA, RNA or proteins, these macromolecules are transferred from the gel to the membrane, because this membrane is a better support, and the gel is actually a fragile, gel is usually fragile. So what we do is we transfer the fragments onto a membrane, and then on the membrane we allow several treatments so that the membrane, the probe DNA can hybridize with the DNA of our interest, and then finally can be visualized. So this part is transfer to the membrane. What is a membrane? What are different types of membranes? We are going to discuss. But before going to that, just to give you a brief overview. So this is our transfer setup. This is a typical transfer setup. And what we do here is that this transfer is usually done by capillary action. So this is actually capillary transfer. This is actually capillary transfer. So what is being done here is that this gel, so this black one is the gel, right? And the membrane is placed above the gel, just above the gel this membrane is placed, and then upon that some weight is given, some paper towels and some weight is given, and then this setup is kept in a buffer. So what will happen, by capillary action the fragments from the gel will go towards the membrane, so will be transferred into the membrane. And then after this transfer process is complete, then this is a membrane that we get. Then the next step will be hybridization. One step that is necessary in between, that is denaturation. Now what is denaturation? So basically to convert the double-stranded DNA to single-stranded DNA, right? So denaturation is important because in the next step what we will see, that this particular DNA of our interest will be bound or will be hybridized with a specific nucleic acid probe. Now for this hybridization to occur, you know that these two DNA pieces, they have to be single stranded, because if they, the DNA sample that is there on the membrane, if that is double stranded, then of course this process is not foolproof, because this process will not occur actually, because the hybridization will not be is not possible in that case, right? So which is why there has to be denaturation, which is usually done with hydrochloric acid. And then after denaturation, then the next step will be hybridization. So hybridization is, so suppose we have this particular DNA of interest, right? So hybridization is actually done with a probe DNA. So this red one is a probe DNA, Now this probe is designed, it has to be designed in every experiment, and it is designed such that this probe is complementary to the DNA of our interest, the DNA that we want to detect, right? So this probe DNA is complementary to the DNA that we want to detect. Now what we will do is we will allow this probe DNA to be hybridized with the DNA sample that is on the membrane, right? And then this probe DNA will be hybridizing to the specific DNA of our interest, right? So this is how you can see the probe DNA will actually be treated onto the membrane for hybridization, and after hybridization you can see that this probe DNA has hybridized to the places, to the DNA fragments where which is our DNA of interest, or the required DNA that we want to see. Now then, therefore, how will it visualize it? How do we know that where our probe DNA has been hybridized? So for that we have to depend on the label that is there in the probe DNA. So this label can be a radioactive label. So this probe DNA can be labeled with radioisotope. See, this radioisotope is usually gamma 32P ATP. So actually this DNA is labeled with gamma 32 phosphate, right, which is a radioisotope. And if this particular probe DNA is labeled with this radioisotope, then after this probe DNA hybridizes to the membrane and to the specific DNA of our interest, then this image can be developed by autoradiography, right? So this we will keep this membrane onto a film where this autoradiograph image will develop, and we can visualize it, right? So this is an X-ray film where we will visualize our membrane, or the places where the probe DNA has been hybridized, and from that we will be able to detect whether our DNA of interest is present in the sample, in how many fragments, etc. Several details we can reveal from the Southern blot experiment. Now these membranes that I am talking about, these membranes can be of several types. The mostly used membranes are nitrocellulose and PVDF membrane. So this is the structure of a nitrocellulose membrane, and you can see that this nitrocellulose membrane is positively charged, it is heavily positively charged, right? So this nitrocellulose membrane is heavily positively charged. So of course the negatively charged DNA molecules, they will tightly bind to this positively charged nitrocellulose membrane. Whereas in case of, this is polyvinyl diethylene fluoride, PVDF, okay, this is another membrane which is also very commonly used, and it is a better membrane than nitrocellulose. And here also you can see that there are these fluorine molecules which have this delta negative charge. So carbon has this delta positive charge, and the DNA molecules as well as the protein molecules can be transferred easily on these membranes, right? Okay. And the next thing is this probe DNA and hybridization. So to give you a very brief detail of how to do this hybridization. So suppose these are several DNA fragments, okay? These are several DNA fragments. These are the several DNA fragments that have come from our restriction digestion, right? So from the when we digest our genome with these restriction endonucleases, many such fragments will arise, but all such fragments are not the fragment of our interest, or not the DNA of our interest, right? So these fragments will be separated on the gel according to their molecular weight, and these fragments will be transferred to the membrane. So this is a membrane, right? So these fragments will also be transferred to the membrane. Now there will be hybridization step where we will have a probe DNA, and this probe here we can see that it is labeled with 32 phosphate, which is a radio label or radio isotope, right? So radioactive isotope of phosphate, and this 32 phosphate probe, this is a particular DNA, right? So this is the DNA which is complementary to the sequence that we want to actually check, right? So for this, what information do you need? The information that you will need is the sequence of the DNA of interest, right? So the DNA of interest that we want to see, its sequence has to be known, and the probe DNA has to be designed such that it is complementary to this particular sequence, right? So it has to be complementary to this particular sequence, and also this probe DNA has to be labeled. It can be 32P labeled, it can also be fluorescent labeled, right? So there will be different types of labeling, but this probe DNA has to be labeled so that we can visualize the probe DNA after hybridization, right? Because the DNA fragments that we are getting from the sample, those fragments we will never be able to visualize, but we will only be able to visualize the fragment where the probe DNA will hybridize, right? So which is why the probe DNA has to be labeled either with a radioisotope or with a fluorescence label. Now here, this particular black one is this probe DNA, and after we treat the membrane with this probe DNA, what we see that this probe DNA will obviously go and bind to its complementary sequence, right? And which is why I told you that these sequences has to be single stranded, right? So this DNA has to be single stranded, because you know, so suppose this is one particular DNA fragment, and if another DNA fragment has to anneal to it or hybridize to it, so this also has to be single stranded, and the probe also has to be single stranded, so that they can base pair with each other, right? So therefore it is essential for the fragments to be single stranded and also the probe DNA to be single stranded, right? So this probe DNA will hybridize to the fragment of our interest, and then what we will do is we will expose the membrane to X-ray film, and where this image will develop, where we can see that the probe DNA will hybridize, and we will be able to visualize the probe DNA, the signal that is coming from the probe DNA. Now after we can visualize such a band in the image, we will be able to confirm that yes, the sequence of our interest which we want to visualize, it was present on this particular sample, right? Or in this genome of the particular organism, this sequence was present, which is why we have got a signal after southern blot. So that is the overview of this experiment, southern blot. What are the applications? So as I mentioned, gene discovery, gene mapping. So whether a gene is present in one organism or not, and the mapping of the genes, this can be done with southern blot. Identification of transferred gene in transgenic organism. So suppose you are transferring a particular gene in a organism by some methods, by some recombinant DNA technology methods, and then after that we want to confirm whether this gene is actually transferred or not. right? So for that you can use this southern blot experiment, and you can design a probe which will be complementary to the transferred gene sequence, right? So after that what you can visualize is that whether this gene is, or whether this probe DNA is hybridizing to the gene sequence or not, whether there is a signal that is coming from the probe DNA. So then we can confirm that yes, this gene has been transferred in the transgenic organism, which is why the probe has hybridized to it and got a signal, right? And also this there are immense applications of southern blot in diagnostics and also in forensic studies. So any doubts so far? Yes, tell me. If you have any doubt, you can unmute your mic and you can also tell me. Okay, alright, then we will move on to the next immunoassay technique, which is called the northern blot. Now as Edwin Southern, he discovered this particular technique, the southern blot, for identification of DNA sequence. Later on these techniques were developed where the RNA or the macromolecules like RNA and proteins can be detected by several techniques, and by following this particular trend, these techniques were named as northern blot for detection of specific RNA sequence and western blot for detection of proteins, specific proteins. So northern blot is a very similar technique to the southern blot, which is used for detection of RNA sequence, right? So RNA is the macromolecule that we want to detect. So what we do here is that we, similar to southern blot, we have a sample and from which we extract the RNA. So RNA is extracted from it, and then RNA is run on gel. So gel electrophoresis is done on agarose gel, and then this RNA samples, they are transferred onto our membrane, which is usually a PVDF or nitrocellulose membrane, right? And then here again what we see that the next step will be the hybridization with the label probes, right? So the label probes can bind to the RNA fragments of our interest, RNA of our interest, and then these RNA sequences can be visualized on X-ray film. So if the label probes are radio labeled, right? So they are radioisotope labeled. So which is a very similar protocol as that of southern blot, but the difference between these two is that southern blot is used to detect specific DNA sequence, whereas northern blot is used to detect specific RNA sequence. And here there will not be any denaturation step because RNA is single stranded, so we do not need to denature the RNA sample as that of southern blot. So what are the applications of northern blot? So a very crucial application of this northern blot is to study gene expression at mRNA level. So we know that we know the central dogma, right? So we know that from DNA, messenger RNAs are formed by transcription, and messenger RNAs transfer their information to form proteins, right, which are the functional molecules of the cell, and by a process called translation. So that is the central dogma, we know, right? And the genes that are expressed, so basically the genes, they first form, if they are expressed, right, if this gene is active, then from this gene an mRNA will be synthesized, right, and from the mRNA proteins will be synthesized. Now gene expression can be studied at two levels. One is at the level of mRNAs and another is at the level of proteins. So mRNA means whether the mRNA of this particular gene is being expressed or not, and finally from the mRNA whether the proteins are being expressed or not. Because if even if the mRNA is present, we are not sure that from the mRNA proteins will be expressed definitely. That is not a certain event. So we need to check gene expression at both levels, that is at the mRNA level and that is at the and also at the protein levels. So for studying gene expression at the mRNA level, northern blot is a very good application, because is a very good method, because we can visualize whether the mRNA which is corresponding to the gene of our interest, whether that is being expressed, and if that is expressed, we will get a signal in the northern blot. Also to study the size of the mRNA, how much is the molecular weight of the mRNA, how much is the size of this particular mRNA which is expressed, and also to study the RNA splicing event and RNA half-life. So splicing, you know that different sizes of the mRNA are actually expressed because of some alternative splicing events, right? And splicing is where the introns are excised out, right, and the exons are joined together. So whether the introns are being retained in the mRNA or not, and whether there is alternative splicing, those things we can study by using this particular experiment, northern blot. Right, so far if it is clear, then we will move on to the last immunoassay technique, which is the Western blot. Now we are going to discuss Western blot in a little bit more detail, because this is much different than the previous two techniques, the Southern blot and Northern blot. Here what we want to detect, we will detect specific proteins. And there are four major steps in Western blot. The first one is separation. So what we do is we suppose we have a scenario where we have a particular cell type. Suppose this cell type is HeLa cells, right? We are growing these HeLa cells, and we want to check whether protein X, some protein, is being expressed in these HeLa cells or not. So we want to check whether protein X is expressed in HeLa cells. So what we will do first, we will isolate the HeLa cells and we will lyse the cells to extract the proteins, right? So that will be the first step. So extraction of proteins is done from these particular cells, and these protein sample, this pool of proteins will actually contain many proteins from the cell. and from that we have to detect whether the protein X is present in this pool of proteins or not. So the first thing that we will do, we will separate these pool of proteins, right? How? By the help of their molecular weight, and of course by using SDS page, right? So SDS page we also have discussed about it, and we know that SDS page is a denaturing page, so we use SDS, and the proteins are all negatively charged because of the use of SDS. So SDS makes all the proteins negatively charged and also uniformly negatively charged, right? So which means that the smaller proteins and the larger proteins, so all these proteins are uniformly negatively charged with the help of SDS. So then these proteins are first of all they are denatured, right, and so they become linear, right, and these denatured proteins and also negatively charged proteins are run on the polyacrylamide gel, right, polyacrylamide gel, and these proteins will be separated according to their molecular weight. So this is a separation that we can see here, the separation phase. So that is the first step. So next step, very similar to the other blotting techniques, will be to transfer these protein samples to a membrane, right? So the next step is transfer. So for transfer, very similar setup we are using here. So we have kept the gel, and upon that we have kept the PVDF or the nitrocellulose. Upon that we have kept the PVDF or the nitrocellulose membrane, right, and on top of below the gel and on top of the membrane we have kept some filter papers, and we have kept some sponges, and we place this particular setup inside a cassette. So there is a cassette and we place it inside the cassette. Now here we do not use capillary transfer. So capillary transfer actually takes a lot of time. So what we use is electrotransfer. So we use electric current to allow the transfer of the proteins from the gel to the membrane, right? How we transfer these proteins from the gel to the membrane, we will see later on. But so this is the transfer process where the proteins are transferred from the gel to the PVDF membrane. Now the next step will be staining. So now after the proteins are already present on the membrane, the next step will be to visualize the protein of our interest. For visualizing the protein of our interest, so suppose this orange one is our protein X, which we want to visualize, and it is bound to the membrane. So suppose here is a membrane, here is a membrane, and the antigen, or in our case this protein of interest, is present on the membrane. Now the this particular protein will be detected by an antibody. So why we are using antibody? Because antibodies are very specific for the antigen. So if we design an antibody against our protein of interest, so suppose we have this protein X and we have an antibody which is designed against protein X, so anti-protein X antibody or anti-X antibody, right? So this protein, if it is a commonly available protein, so usually we have many antibodies which are designed for our proteins, so many specific proteins, and we can purchase that antibody also. Or if the antibody is not available, then what we have to do is we have to raise an antibody. So that is a separate technique, so where we have to inject the protein X into a organism, which is usually mouse or rabbit, and then we have to collect the serum of this organism, the mouse or the rabbit, and then from the serum we can get this anti-protein X antibody. Now this anti-protein X antibody, why is it used? Because it is very specific, it will be very specific to protein X, and wherever protein X will be bound to the membrane, the particular antibody will be able to recognize this protein X and it will be able to bind to it. Now this is actually the primary antibody or 1 degree antibody, it is said. Primary antibody or 1 degree antibody. So primary, why is it called primary antibody? Because this directly detects the antigen of our interest or the protein of our interest. Now it is very important to mention here that we can see there are two antibodies which are used. There are two antibodies which are used. One is a primary antibody, another is a secondary antibody. And secondary antibody is a special type of antibody because it is conjugated with an enzyme. Now why is it conjugated with an enzyme? For the detection purpose, right? So this enzyme is either it will be enzyme which can give a colored product or which can give a chemiluminescence. So there are certain enzymes which are conjugated with these secondary antibodies, so that when we treat this particular protein with a substrate, or treat this membrane with a substrate, if this enzyme is present, then this substrate will be converted to the product by this enzyme, and this product will give a certain signal. It can be colorimetric, it can be chemiluminescence, it can be different types of signal which will be given by the enzyme. So this secondary antibody is used so that now this secondary antibody can recognize the constant region of the primary antibody. So this was our primary antibody, the anti-protein X antibody, which directly recognizes the protein X. Now this primary antibody is not labeled. This primary antibody is unlabeled, but the secondary antibody that we will use will actually be recognize the FC region or the constant region of the primary antibody. So the anti-primary antibody will actually be our secondary antibody. So the constant region, so if you have any idea about the antibody structure, the antibody has a variable region and this part is the constant region. The constant region of the antibody is recognized by a secondary antibody. Now these constant regions are actually species specific, which means that suppose the primary antibody is raised in mouse, okay, so all the antibodies of the mouse will have a same constant region, more or less. It can have same constant region. So this constant region will be detected by the secondary antibody. So this secondary antibody will actually be an anti-mouse antibody if the primary antibody is raised in mouse. So are you getting my point? So the thing is, first of all, the primary antibody has to be raised in an organism. It can be mouse, it can be rabbit, it can be donkey, it can be any other organism. And then the secondary antibody, which will have to recognize the primary antibody, will be raised in another organism, and it will be able to detect the anti-mouse or it will be able to detect the mouse constant region. So which is why it will be anti-mouse antibody. And also this secondary antibody will be labeled with an enzyme which can give a colorimetric signal or a chemiluminescent signal or a fluorescent signal. So that is why we are using two antibodies here. So you might ask the questions, why the primary antibody is not directly labeled with an enzyme? Why are we using a secondary antibody? Because it is not very cost, so it's very expensive to actually label all primary antibodies with enzyme. So which is why we have fixed secondary antibodies which are labeled with enzyme. Now suppose we have our primary antibody raised in rabbit. So what we do is we purchase a secondary antibody which is anti-rabbit conjugated with enzyme, right? If our primary antibody is raised in mouse, we purchase an anti-mouse secondary antibody conjugated with enzyme. Or if our primary antibody is raised in some other species, so our secondary antibody has to be specific to that particular species, right? It can be rat, it can be mouse, it can be rabbit, it can be donkey, it can be any other organism as well, right? So which is why we do not have to specifically label all primary antibodies with enzyme, which becomes very expensive. So that is the first point, why we use a secondary antibody which is actually fixed and which can recognize the FC region of our primary antibody. And also we use a secondary antibody because it amplifies the signal. So for signal amplification, and because it is it's not very expensive, so it reduces the cost, which is why we use this secondary, like two antibody system. And here this enzyme, we will discuss later on what can be the enzymes and how they will give us the signal. Now after that, the next thing will be to develop the signal, and we can visualize this. So the last step is the visualization step, and we will be able to visualize the bands where the protein of our interest we can visualize, and the bands that will form will actually be the bands or the proteins where the primary and the secondary antibody has bound and given a signal, right? And this will be the band of our interest or the protein of our interest. So this will be, suppose, protein X, right? So that is the visualization step. Any doubts so far? Okay, so now we will discuss these steps in a little bit more detail. So first step is the separation by SDS page. So we will run the protein samples in the gel. And we will allow the negatively charged proteins to be separated on the polyacrylamide gel. And so from the cathode towards the anode, the proteins will migrate, because the proteins are negatively charged, so they will migrate towards the anode, right? And we will also use here, so you can see this thing, this is actually a pre-stained protein ladder or pre-stained protein marker. why do we use a pre-stained protein marker? So it looks like this. So this is a pre-stained protein marker. So you can see that this protein marker has obviously you know that protein markers have specific proteins of specific molecular weight, and when they are run in the gel and they separate from their molecular weight, we can have an estimate of the molecular weight of our sample, right? And why do we use a pre-stained protein marker here? Because when this when the contents of these gel, when they will be transferred onto the membrane, so the proteins, they are not labeled, they are not they do not have any color. So when they are transferred from the gel to the membrane, we will not be able to visualize them because the membrane will be white, simple. So that is why we use a pre-stained protein marker, because this pre-stained protein marker has different colors, like this green, red, so and the blue ones, so they have different colors. So these protein markers will be visualized, we can visualize them in naked eye when they are running in the gel. So they will form these colors, and also when this gel will be transferred to the blot, so this is the gel and this is on the blot. So when these proteins will be transferred onto the blot, on the blot also we will be able to see this marker, right? On the blot also we will be able to see these marker bands, so by which we can have an idea that yes, the protein samples from the gel has been transferred to the membrane, because the marker has also been transferred, right? So this is a good indicator of whether our transfer process was was correctly done or not, which is why we are using a pre-stained protein marker here. And so at the end of this separation, this is an SDS page, and here what we do is that we do not stain with Kumasi blue. So this Kumasi blue staining, it is not done before the blot, okay, because we will be transferring these proteins to the membrane, which is why we do not stain with Kumasi blue. After this SDS page is run, we take the gel and then we directly transfer the proteins onto the blot. Now this is the next step, that is the blotting and transfer. So this is a setup that we can use for transfer, and here you can see that there is this this transfer apparatus, right? And in this transfer apparatus there is this cassette. So this cassette has electrodes. So I told you that this is an electrode transfer method, where the proteins, these are negatively charged. So if you remember that proteins, they are charged with SDS, right? So which is why they are negatively charged, and these negatively charged proteins will move towards the anode, which is the positive electrode. Right, so when these proteins, negatively charged proteins, will move towards the anode, and if we place the membrane towards the anode, then the proteins from the gel will be transferred towards the membrane, right? So the gel and the membrane, so proteins are transferred from the gel towards the membrane. So membrane is kept at the anode, membrane is kept towards the anode or the positive electrode, because the proteins are negatively charged. So the proteins will move from the gel towards the anode because it is positively charged, and the gel is placed towards the cathode. So gel is actually, so the proteins are negatively charged, so they can move in the opposite direction, they can move towards the positively charged electrode. So in this process they will be transferred from the gel to the membrane. If we by mistake alter this position of gel and the membrane, then the transfer will not occur. Okay, so that will be a mistake and the transfer will not occur. The proteins will not be able to move towards the negative electrode, So if we place the membrane towards the negative electrode, the transfer will not occur. And so this gel and the membrane, they are placed on top of each other, and on top of that we are placing some weight by the filter papers, by some sponge, and then we are placing this this total system onto this cassette, right? And then this cassette is placed in this tank, right? And then we pass electric voltage through it so that the proteins can be transferred from the gel to the membrane. And here the transfer buffer, there we have to use a transfer buffer, which usually the contents of transfer buffer is tris, glycine and methanol. Tris, glycine and methanol. And this is how the transfer is done. Now we have to remember certain things while we are transferring the proteins. That is, this transfer process is usually done at low temperature. So in a refrigerated chamber, this transfer process is usually done, or there has to be a cooling unit. We can see here this cooling unit has to be done so that this actually cools down the whole system. Why is it used? Because when the transfer will occur by the passage of the electric voltage, so a lot of heat will be generated, and if this heat is generated and if the system temperature goes up, the proteins will not be properly transferred, the proteins might degrade, which is why the transfer has to be done at lower temperature in a refrigerated chamber. And there is also a possibility that between the gel and the membrane there might be some bubbles. So if there are some bubbles, some air inside the in between the gel and the membrane, then this transfer process will be hampered. The electric voltage transfer will not be efficient because of this presence of air inside or between both of these. So which is why the gel is placed, and on top of that this transfer membrane is placed, the membrane is placed, and we make sure that we roll something on the membrane. We use a roller to roll on the membrane so that there should not be any air space or any bubble present in between the gel and the membrane. Because if there is bubble, we will see that there will be some white spots that will appear on the membrane after the detection. So after visualization, we will be seeing some white spots which will actually hamper our visualization. So that has to be taken care of while we are doing the transfer. Next step is blocking. It is very essential to block the membrane before we start any antibody treatment. So what is blocking? So suppose this is our membrane where these blue triangles, these are protein X. So this is our protein of interest, right? And there will be some other proteins also. Because from the cell lysis we have got many proteins in our sample. Now the rest of the space on the membrane is free. So like this part, it's free, right? This part of the membrane, it is free, but this membrane is a charged membrane. So when we will treat the membrane with antibodies for staining, for detection, then these antibodies can bind to these empty spaces on the membrane and can give non-specific signals. So to prevent that, what we have to do is we have to minimize the empty spaces on the membrane by non-specifically saturating the membrane with some protein, like for example bovine serum albumin, For example, bovine serum albumin, BSA, casein, gelatin, or a non-ionic detergent like Twin 20 can also be used for blocking, and skimmed milk. So non-fat dry powdered milk, it is also a good blocking reagent and economic blocking reagent also. So this membrane is saturated with this kind of protein samples. So these proteins will non-specifically coat the membrane. So suppose this is BSA, and this will coat the empty spaces on the membrane, right? So that the protein, the antibody that we will be using in the next step, these antibodies, they can only bind to the protein of our interest and do not bind to these empty spaces, right? This binding is minimized with the help of blocking. So blocking is a very, very crucial step in Western blot, because if we do not block the membrane properly, it will give us many non-specific signals and we will not be able to be certain about the presence of our protein of interest, right? So this blocking can be done for, actually the blocking can coat our antigen of interest also, which is why after blocking we have to rigorously wash the membrane. So blocking is followed by washing. So washing is done with buffers like TBS, Tris buffer saline. So why is TBS used? Because, or why is this washing step important? Because after blocking we will allow this buffer to wash the membrane, so that all the excess blocking reagents, they can be removed from the membrane, right? So the excess blocking reagents should be removed before we treat with antibodies. So this is also very, very important in Western blot, that is to wash the membrane. After blocking, after antibody incubation, in various steps, the membrane should be washed properly, otherwise many excess reagents will be present which will hamper the process of visualization, right? So blocking has to be done, and after that washing also has to be done. This blocking can be carried out at lower temperatures like 4 degrees centigrade, or this blocking can be carried out at room temperature also. So that time of blocking, for how long we will block the membrane, this time varies, and this has to be standardized as per our experiment. Then followed by blocking, we have to rigorously wash the membrane so that all the excess blocking reagents can be removed and the antigens can remain free on the surface, right? So after that, what we will do is we will treat the membrane with antibody. So this is the membrane, we can see. No, so Twin 20 we use usually for actually a very less concentration of very low concentration of Twin 20 is used, so it is not harmful for the membrane, for the protein of our interest. And anyway, the proteins that are bound to the membrane, the proteins are all denatured proteins, right? So Twin 20 will not be harmful, and we use a very low concentration. Now there is a membrane on top of which, suppose this is our protein of interest, this particular one is our protein of interest, right? And first what we will do is we will treat the membrane with a primary antibody solution. So we will dissolve the primary antibody in a particular dilution. Now this dilution of the antibodies are also critical for proper visualization. So this antibody dilution can be 1:500, 1:5000. So it depends on the type of antibody that we are using and on the specific protein that we are trying to detect. On depending on that we can use different dilutions of antibodies, 1:10,000, 1:20,000. It can be varied, and it can also depend on whether the antibody is monoclonal antibody, whether the antibody is polyclonal, and so on. So first what we will do is we will make a primary antibody dilution, and then we will soak the membrane in this primary antibody dilution, and we will keep it on a rocker so that we will keep it in a box, and then the box will rock gently so that the primary antibody can get in contact with the target membrane, and it can bind to the antigen of our interest or the protein of our interest. So you can see this primary antibody binds to the antigen of our interest. Now after this primary antibody incubation is done, then this solution is taken out, right? This primary antibody solution is taken out, and the membrane is again washed. Excuse me for a minute. So in between this primary antibody and secondary antibody treatment, there also has to be a washing step. So where we wash off the excess primary antibody, because if we do not wash the excess primary antibody, it will again give non-specific signal. So in between these two antibody treatments, there has to be washing step. So then we wash the membrane, and then we again treat the membrane or soak the membrane with secondary antibody solution. So this secondary antibody solution is usually given in a much more dilution, so it is more diluted than the primary antibody solution, and the secondary antibody is also conjugated with an enzyme. Now here there are two enzymes mentioned here. One is horseradish peroxidase, HRP. Horseradish peroxidase is actually a peroxidase which is extracted from this plant, horseradish. So this is a plant, it's not horse, it is a plant, radish plant, from where this HRP enzyme is extracted. So it is it gives chemiluminescent signal. So the substrate of horseradish peroxidase is H2O2 and luminol. H2O2 and luminol. So this peroxidase enzyme can oxidize H2O2, and that will emit a chemiluminescent signal from the luminol substrate. So that is how we can detect so when we treat the membrane after these antibody treatments are done, we again wash the membrane with this wash buffer, right? So after these two antibody treatments, we are washing the membrane, and then we are treating the membrane with an enzyme, with a substrate. So this enzyme is already conjugated to the secondary antibody. We are treating the membrane now with a substrate, which is H2O2 and luminol in case of HRP, and wherever this particular enzyme is conjugated with the secondary antibody, and wherever the secondary antibody is bound in the membrane, then H2O2 and luminol will actually give the chemiluminescent signal, and this chemiluminescent signal can be detected by a chemiluminescence detector. There can be another enzyme which is commonly conjugated with secondary antibody, that is AP, alkaline phosphatase. This is an enzyme which converts paranitrophenyl phosphate to paranitrophenol, and paranitrophenol gives yellow color. So this is a colorimetric detection. This is colorimetric detection. whereas this Horstedis peroxidase is used for chemiluminescent detection. So that is the role of this secondary antibody, which is conjugated with enzyme. And here we might often see in the blots that we do not see any signal, we do not see any bands. This absence of bands can be due to many reasons. One reason can be obviously the protein is not present. One reason can be that the blot, the experiment has not worked, and this can result from various reasons, like for example, if the enzyme which is conjugated with the secondary antibody, if that is not functional. So suppose this antibody is old, it's not fresh, and because of that what might happen, the enzyme might not be functional. So if the enzyme is not functional, even if there is a protein which is present, the protein of interest is present on the membrane and it has been detected by the primary antibody, and the primary antibody has also been detected by the secondary antibody, then also if this particular enzyme does not work, then we will not see any signal, either colorimetric or chemiluminescence. So that enzyme has to be functional, so that there has to be that develops a signal. So these things are required to be taken care of while we are doing a Western blot experiment. Now, what are the applications of Western blot? First of all, we can determine the size and the amount of protein in our given sample. So whether a protein is expressed in very small amount in the sample, in the cell, or whether it is expressed in large amounts in the cell, we can detect that by Western blot. So Western blot is a semi-quantitative technique, we can say, because from the intensity of the band we can have an assumption that whether the protein is present in large amount or in small amount, and also the size of the protein. So we are running a protein ladder on the gel, which will also be transferred on the blot, and from the size of the protein ladder we will be able to comment on the size of our protein of interest, right? So we will be able to determine the size of our protein of interest. Now Western blot is very commonly used for detection of many diseases, like it can detect the antibodies against virus or pathogen in our blood serum. So suppose we are infected with a particular virus, right, or a bacteria, so our body, our immune system will have to fight that bacteria or pathogen or virus, and for that our immune system will develop antibodies against that pathogen. So these antibodies will be present in our blood serum, right? And if we collect our blood serum and if we do a Western blot experiment on that, and we detect the antibody with again another antibody which is specific to that particular antibody, so by that we will be able to comment whether or we will be able to observe whether that antibody against the virus or the pathogen is present in our blood serum or not, and by that we will be able to confirm that yes, we have been infected with this virus or pathogen, which is why we have this antibody against the virus in our blood serum, right? So that is a way of confirming about an infection. Western blot is also a confirmatory test for HIV, so human immunodeficiency virus, right? So we have this anti-HIV antibody which can detect the presence of this HIV in our body. And so we know that for HIV the principal way of detection is ELISA. So the first experiment that has to be done is ELISA, but after that if we get a positive signal in ELISA, we have to confirm it by Western blot. So for which there is that is Western blot is used, and also Western blot can be used for detection of defective proteins, like for example in Prions disease. So Prions disease is a disease which is due to some misfolded proteins which forms aggregates, so that can be also detected by Western blot. So Western blot has a very wide applications in detection of various diseases as well as in laboratory purpose, so for detection of whether the protein is expressed in a particular cell and what is the size of the protein, whether this protein is expressed in particular stress conditions, those things can be studied by Western blot. And two things that I would like to mention here, that Western blot, getting a good blot, so this particular image is a good blot. This is an image where we can see that the bands that have formed, they are crisp bands, right? So getting a good blot is actually difficult. It's not very easy. This seemed like an easy experiment, but getting a good image out of it is not so easy. So there might arise different problems. So these are such plots where there were many problems. So here in this particular blot you can see that there is a blotch that appeared on the blot, so it's blotchy. Here we can see that it's totally dark, so it has very high background. These bands indicated with these arrows, these are ghost bands. These are called the ghost bands. So you can see that there is a dark outer line of the band, and in between the band is white, not like this, right? These bands are called ghost bands. Then these bands, these are called smiley bands. So forms like a smiley kind of a structure, right? Not crisp like this, right? There is a drag in the blot, we can see. Here we can see there are many white patches on the blot. So these problems might arise in Western blot, and we have to know how to get rid of these problems. So the few points that we have to take care, or we have to remember while doing an Western blot experiment, is that the protein which is loaded in the gel, the protein sample that is loaded in the gel, that has to be standardized. So the protein amount, if it is too low, then we will get no signal, right? Because if it is very low for being detected by an antibody, then we will not get any signal. If it is very high, then we will get these ghost bands kind of things, or we can get blotches also on the band, on the blot. We can get blotch, we can get ghost bands, or if the amount of protein is very high on the gel, so it forms a what we'll say, a kind of a blotch will appear, because a lot of antibodies will bind to this protein band, and it will give a blotchy signal, right? So that signal will not be crisp, the band will not be crisp. Now the next thing is blocking. So blocking is a crucial step, and the time for which we are blocking the membrane also has to be standardized. It can be done overnight, it can be done for 1 hour also. So depending on the experiment that we are doing, the duration of blocking has to be standardized, because if the blocking is done for less amount of time, then there can be non-specific signals that can arise. If the blocking is done for a very long time, then there can also be background that can develop in the blot. So the whole background will be black, that kind of thing. And the membrane also has to be properly washed after this blocking and antibody incubation. Otherwise there will be non-specific signals. So this washing is also very critical. And the other thing is the dilution of the antibody, the incubation time of the antibody. These things has to be also standardized. So in which dilution we are using the antibody. if it is too low, if it is very diluted, then it will not be able to detect the protein of our interest. If it is also very high, then it will give a background signal, it will give non-specific signal. And the incubation time, so for how long we are incubating the membrane with the antibodies, that also has to be standardized according to the experiment. And the antibody should not be cross-reactive, which means that suppose our secondary antibody, the secondary antibody should only be able to detect the primary antibody. So the secondary antibody should only be anti the Fc region of primary antibody, anti-Fc of the primary antibody. So the secondary antibody, it should only be able to detect the constant region of the primary antibody and nothing else. If it detects some other proteins on the blot, on the membrane, then it will give signals which we are not expecting, right? So we only want the secondary antibody to react with the primary antibody, to bind to the primary antibody constant region and not cross react anywhere else on the blot. And also the secondary antibody has to be carefully chosen. So if the primary antibody, I am again telling this, if the primary antibody is raised in rabbit, then the secondary antibody will be an anti-rabbit antibody which is conjugated to an enzyme. And if it is an anti-rabbit antibody, it cannot be raised in rabbit, right? So it has to be raised in some other organism, like for example mouse or rat, right? So the two antibodies has to be raised in two different organisms. One, if it is raised in rabbit, the other one will be anti-rabbit but not raised in rabbit, of course. Because if it is raised in rabbit, then it will cross react with all the proteins of the rabbit organism, so rabbit cells. So therefore this particular anti-rabbit antibody has to be raised in other organism like mouse or rat, right? So these things have to be carefully chosen, and which will actually help us to get a good image of Western blot. So any doubts so far? Alright, so we will move on to the questions. Okay, thank you. Let's move on to the questions. Let me know your answers. Correct answer. Next. Correct answer. Next question. Correct. Next question. Yes, correct. Immunoblotting. Next question. Correct answer. Next question. Correct answer. Next question. Very good. Correct answer. Next question. It is the powder milk. So it will be actually powder milk and 220 both. But powder milk is a very good blocking reagent. Next question. Good. Right answer. Next. Correct. Next. Correct answer. Next. Correct answer. Next question. Yes, PVTF is a good answer, good option for stripping a membrane. So nitrocellulose is also a good membrane, but for stripping, so which means that suppose you have used a membrane and you want to reuse it, so or you have run the you have transferred the samples on the membrane, and then what you want to do is you want to remove the antibodies from the membrane that were used for detection, and then you want to incubate the membrane with again another set of antibodies. For that, PVTF is a very good option. Next question. So actually all of these problems might arise. If washing is not done correctly, background noise will also be high, which is why the whole blot might be dark, and if there is background noise, no specific band can be observed. So actually the correct answer is all of the above. And the last question. Yes, C is correct, but also B. B is also correct, because if there has been too much of protein, then these ghost bands might also arise. So both B and C are the correct options. Okay. So that's all for today. Thank you for joining. If you want some additional information on Western blot, you can check this link. This is a video I had made to for a detailed description of Western blot, what to do and how to run a good Western blot. And thank you for joining. If you have any other doubts, please let me know. So this is the last session today. With this, we are ending the experimental biochemistry like sessions. Thank you, and all the best for your exams.